So, do you all have any idea about what are HLAs? Like, what is the full form of HLA? Human leukocyte antigens. Okay. So, now coming to HLA, so we have few molecules called the major histocompatibility complex molecules. Okay. So, why are they important? Why are these complex molecules important? Like it helps in determining the compatibility between individuals. Okay. So these MHC molecules, which is the short form of major histocompatibility complex, these MHC molecules in humans are known as HLA molecules. Okay. So they are human leukocyte antigens. So what is the function? They bind peptide fragments of foreign proteins for presentation to antigen specific T cells. Okay. So now these antigens, they are responsible for graft acceptance or rejection. So if the HLA is not matched properly, then there will be a rejection, okay, when we do a transplant. And these HLAs, they are also responsible for many autoimmune disorders, okay. So that's why it is very important. So now, what is HLA haplotype? It is the combination of HLA alleles in each individual, okay. So these HLA haplotypes are never same in two individuals except identical twins, okay. So everyone will have different HLA haplotypes. So now coming to the function, they display peptide fragments of protein antigens for recognition by antigen specific T cells, and in humans these MHC molecules are called human leukocyte antigens, which I have already told you all before. So coming to the location, so these HLA molecules, they are located in the short arm of chromosome number 6. Okay. So now these encode some genes, okay. So these HLA molecules are classified into 3 types based on the structure, the cellular distribution and the function. So they are of 3 types, class 1, class 2 and class 3. So the class 1 molecules encode for 3 genes, that is HLA-A, B and C. The class 2 molecules, they encode for a region called HLA-D, which has again 3 subregions, okay. So these 3 subregions are HLA-DR, DQ and DP. And class 3 molecules, they are components of the complement system. So that will be dealt with you all later, okay. So now as you can see, this is the chromosome number 6. And in the short arm of the chromosome number 6, you get these HLA molecules, okay. So now this HLA system is highly polymorphic. That is, no 2 individuals are likely to express the same MHC molecules except identical twins. Then what are these alleles? They are alternative form of a gene at a single locus. There are many alleles of each MHC gene in a population, and each individual inherits one set of these alleles. And so this HLA is a major barrier in organ transplantation. Jeta ami bollam je in organ transplantation HLA matching is must, okay, or else the patient will have a rejection. So now nomenclature. So how are these HLAs named? As I told you all, the HLA, the gene locus is usually this first 4 digits, that is DRB1, means the location. Then the allele number is 04 and order is 02. So for your stage, you don't need to know the whole HLA number. I will tell you all, okay, what all to remember. So this is how the HLAs are named. So now coming to class 1 and class 2 MHC molecules. So what are their structure? So coming to the class 1 MHC molecule, as you all can see, they are heterodimers, okay. So what they have a polymorphic alpha chain and a non-polymorphic beta 2 microglobulin. So as you can see, the class 1 MHC is composed of alpha chain and a beta 2 microglobulin protein over here. So now these alpha chains, they encode for, as I told you all, HLA-A, HLA-B and HLA-C. Now this alpha chain is divided into 3 domains. So what are the domains? Alpha 1, alpha 2 and alpha 3. Can you all understand? Okay, so now this alpha 1 and alpha 2 will form a cleavage, okay. And this site is the peptide binding site, where the peptide is binding, okay. So now this alpha 3 has a binding site for CD8 plus T lymphocytes, okay. So now once the protein binds to this MHC class 1, what will happen? They will present these cells to CD8 plus T cells. Understood? So now these CD8 plus T cells, hence they are known as cytotoxic T lymphocytes, and CD8 plus T cells are also known as MHC class 1 restricted. Why? Because they will only recognize the proteins which are bound to MHC class 1. Did you all understand? So you all might have to draw this diagram, okay, for MHC class 1 and class 2. So this is the structure of MHC class 1 molecule. If you all have any problem, please tell me, okay. So now coming to MHC class 2. So MHC class 2 te kiya chhe? We have polymorphic alpha and beta chains. So this alpha chain will have 2 domains, that is alpha 1 and alpha 2, and the beta chain will also have 2 domains, beta 1 and beta 2. Now here the peptide binding site is between alpha 1 and beta 1. So alpha 1 domain and beta 1 domain will form a cleavage which will be the peptide binding site for MHC class 2 molecules. So now the beta 2 domain of MHC class 2 has a binding site for CD4 plus T lymphocytes. So these CD4 plus T lymphocytes are MHC class 2 restricted, okay. So as you know, like what is MHC class 2 restricted? That is like the proteins which bind to MHC class 2 will only be recognized by CD4 plus T cells, and these CD4 plus T cells are known as helper T cells. So now as I told you all, MHC class 2, they have a special encoding region called HLA-D, which is subdivided into HLA-DP, DQ and DR. So now coming to HLA, the class 1 MHC molecules. So class 1 MHC molecules, where will they be present? They will be present on all nucleated cells and platelets, okay. So now as I told you all, they are encoded by 3 closely linked loci or genes which are HLA-A, B and C. So their function is they display peptides that are derived from cytoplasmic proteins, including normal proteins. So examples will be viral antigens and tumor specific antigens. ঠিক আছে, বুঝতে পারছো তো? Now coming to class 2 MHC molecules. Now class 2 MHC molecules are coded for a region called HLA-D, and these I told you all, these are situated on antigen presenting cells. Mane the cells will already engulf the extracellular microbes and then they will present it to the CD4 plus T cells, okay. So that's why they are mainly situated in the macrophages dendritic cell and the B lymphocytes. Okay. So they present antigens derived from extracellular microbes and proteins following their internalization into the endosomes or lysosomes. The internalized proteins are proteolytically digested, producing peptides that then associated with class 2 MHCs. ঠিক আছে? Now coming to HLA testing methods, you need not know the details, just remember the names if possible, okay? So first HLA testing methods one is the DNA based typing, which can detect both class 1 and class 2. Then coming to serology, same, it can detect class 1 and class 2 molecules. And third is cellular detection, which is only done for class 2 molecules. So DNA based typing, and what are the advantages? They are specific, that is specificity of each DNA typing reagent is clearly defined and is based on specific known nucleotide system sequence. Then they are flexible, that is testing can be done at several levels of resolution. Then no viable lymphocytes are required, they are highly reproducible, not influenced by any health of the patient. And large scale testing korte hole, they usually do the DNA based typing, which is done for both class 1 and class 2 MHC molecules. And they can detect full extent of HLA diversity. ठीक है sir? Next, coming to the nucleic acid sequencing. This is direct determination of DNA sequence of HLA alleles. So it is a sequence based typing. Then where is it used? It is used mainly in the bone marrow transplantation. What are the demerits? It is labor intensive, a lot of people are involved, and it is a highly complex procedure, okay? And automated, some sequence based typing methods are HPLC and pyro sequencing. Ato details mone rakha dorkar nei, ekটু just remember the names, like if you are asked you can just write, or in a short note also you can just mention the methods, okay? Then coming to HLA and disease association. So first and foremost, I told you all that HLA is very necessary for especially organ transplantation. Okay, so why do we do HLA testing in organ and bone marrow transplantation? To avoid graft versus host disease, that is GVHD, and to avoid rejection of the graft. Okay. So now HLA alleles, they usually determine the disease susceptibility. As I told you, it is also used in autoimmune diseases, like if a person has a particular HLA, then that person will have a higher risk of developing that autoimmune disease. Okay. So that's why it is used for disease association and to determine the prognosis of a disease. So now in tissue or organ transplantation, what happens? Usually we do this to maximize donor and recipient compatibility and to minimize the impact of immune response on the transplanted organ. So this is why we do an HLA matching. And so what all HLAs are needed to match in an organ transplantation? We usually match the HLA-A, HLA-B and HLA-DR. And individuals who are matched at whatever resolution for all three loci are determined as termed 6 by 6. Mane ei tintei jodi match kore jay recipient আর donor এর মধ্যে, then it is a 6 by 6 match. Okay. Next, coming to HLA specific antibody detection in recipient serum. So here the patients can be sensitized to a specific foreign HLA molecules, like which happens in cases of transfusion, then pregnancy or prior to the transplantations. Mane before doing the transplantation only we are sensitizing the patient so that the immune system doesn't cause a rejection. Okay. So now assessment of compatibility between the recipient and the possible donor includes the testing for his sensitization, and the recipient serum is tested prior to the transplantation on a panel to measure the panel reactive antibodies. Before the transplantation, we usually do this panel to check so that the patient doesn't have any rejection after the procedure. So at the time of transplantation, the recipient's serum is tested with the prospective donor lymphocytes and to identify specific reactivity to the potential donor in donor-specific cross-matching. Now coming to the prerequisites, এগুলো just খুবই superficially তোমরা জানবে যে কি prerequisites লাগে, মানে ডোনেশনের আগে কি করতে হয়, ঠিক আছে? So as in case of renal transplantation, you have to do a donor selection, then ABO compatibility test করতে হবে, then T cell donor specific cross match negative with appropriate recipient sera and best available HLA matched organই তোমাকে নিতে হবে. And for non-renal organ transplantation, মানে kidney বাদ দিয়ে যদি অন্য কোনো transplantation করা হয় তখন HLA typing is not done usually and no pre-transplant donor cross-match is done. And for bone marrow transplantation, usually we take HLA identical sibling or HLA matched unrelated donors. Now coming to disease associations. But disease associations, these are all the autoimmune diseases which are HLA, ঠিক আছে, HLA associated. So just few things you have to remember which is very important for MCQs also. Just remember the HLAs, okay? Otto borrow, you need not remember the whole number, but at least the locus you have to remember, okay? Like HLA DRB1 is associated with what? Okay, so this is also very important for MCQs. So first disease which is associated with HLA is your rheumatoid arthritis. So usually the anti-CCP antibody positive rheumatoid arthritis will be HLA associated, and the primary marker is HLA DRB1. Next, coming to type 1 diabetes mellitus. So here the major determinants of genetic predisposition, like the main HLAs are DQB1, DQA1 and DRB1. Sorry. Then coming to multiple sclerosis. So multiple sclerosis also it is HLA DRB1 associated. I have highlighted it with red color so that you remember only this part. Okay, you need not remember the numbers. ঠিক আছে? Then coming to next disease, which is the celiac disease. Celiac disease is HLA DQA1 and DQB1 associated. Then coming to one more very important group of disorders, that is systemic lupus erythematosus, which is HLA DRB1 associated, and ankylosing spondylitis, which is HLA B27 associated. So this is very important, okay? Like if you are asked that HLA B27 is associated with which disease, you have to say ankylosing spondylitis, okay? And whoever has these HLAs, they will have a 100 to 200 fold increased risk of having this disorder, okay? So now what is transplant associated graft versus host disease? Just in short, you have to know that viable donor T cells, they are recognized and rejected by the host as foreign, okay? Like whenever you put the graft, my immune, like suppose I am having a transplant, so when the graft is put in me, my body will identify it as foreign, so it will reject it, okay? So that get this graft versus host disease. And who are the highly risk patients, like high risk patients who can have this are patients with congenital disorders like DiGeorge syndrome or severe combined immunodeficiency syndrome, which is seen in children, okay? Like they have severe immunodeficiency because of which they cannot accept any grafts, okay? And then patients who are very low birth weight infants, okay? Then Hodgkin's lymphoma patients and patients who need bone marrow transplantations, okay? So there is a high chance of having a rejection. Then patients with normal immunity who are homozygous for an HLA haplotype are also at risk. And donors, they are heterozygous, but they will share one haplotype, means one haplotype needs to be matched. And if you expose these grafts before, like prior to the transplantation, if you expose these to gamma rays or X-rays, then there will be a less chance of the rejection. তাই জন্য অনেক সময় কি হয়, when we transfuse blood, do you know that we irradiate the sample before, like PRBCs or any other like blood samples? Whenever we transfuse, that time we usually irradiate the blood so that any unwanted, these things will, like foreign bodies will be like destroyed, okay, so that it causes less rejection, okay, or any reaction in the patients, okay. So we usually irradiate the samples. So now to summarize, if you get a short note on HLA, will you all be able to write? So what is HLA? Then classification, I told you all the classification. Then the testing methods, the HLA associated diseases, and finally transplant associated graft versus host disease.